Welcome back. Just doing another recording for the second part. Takeaway, Biol3157. So, what are we going to do with all this information? Let me give you a practical example. This is what I've decided to devote my career toward. It's genomic prediction for the environment. We're working on climate, soil, agriculture, and ecosystems and their interconnections. It has been a focus, certainly for agriculture, to improve yield, gain, improve yield year after year, but we're using up our soil fertility. We're requiring more inputs of water and nitrogen. We're expanding in to tropical forests. We're even using our excess food to feed cows and cars, and we got to take a step back, think about not only being more sustainable, and having enough for tomorrow, but also regenerating the ecosystems that we've gone too far. And agricultural yields in many parts of Australia actually declined because we've consumed our soil fertility and groundwater aquifers and depleted. The environment's getting harsher. And biodiversity is at risk. So there's opportunities for linking agro-ecosystems and building soils, and this is a type of agriculture that results in regeneration of fertility, water holding potential, biodiversity, soil microbiology, as well as yield. And that's going to take a little step back. It's going to take a lot of science and integration, but the tools you learned in this course can be applied to this problem. Let's take a look at the soils. So we could also, we did some landscape associations, say rainfall and genotype, but we could also do soil types and phenotypes, and there's these categorical soil types. But what's better really is to look at the chemistry that goes into classifying these soils and associate those variables, amount of limestone, pH, cation exchange capacity, with the genotypes. If natural selection is acting, and certainly seeds that are the wrong genotype won't grow. Some soils won't grow anything much at all, but eucalypts grow across this range, acacia grows across this range, and they must have adapted to these different soil types. So we can get at the genetic basis for that, we can make predictions, we can breed with that, we can conserve and restore and regenerate. It's a whole food web. We know that plants grow into the soil, not out of the soil, and they certainly do take some nutrients up, but mostly it's carbon in, and that requires an association with lots of bacteria and fungi, and the bacteria can fix nitrogen, and the fungi can improve water use efficiency. They can pull phosphorus out of harsh conditions. The soil has a genotype of its own. We're starting to sequence soils now, and we can cluster soils just the same way we clustered host genotypes. We've been thinking about sequencing the rhizosphere, which is the root itself, with all the living things on it. And so half of the genetic reads might align to the host genome. The other half might be novel things that we've never seen before, or they could be mapped to databases of soil microbes. So we can get actually the phenotype of the soil at the same time as we get the genotype. So I'm very excited about that. If you're interested, reach out. We got a lot of projects coming. This was actually at the Paris Agreement. There was a solution proposed. It's called 4P1000. 0.4%, or 4 parts per mil per annum, in the top 30 centimeters of soil. If we could build soil carbon at that rate in our agricultural conditions worldwide, it's certainly possible in some agricultural conditions, not maybe everywhere, but if we could, that would actually cancel out all the anthropogenic carbon emissions. So this is an ambitious target that just kind of got soil on the map. There's going to be a lot of discussion next week in Glasgow about what is possible, and could we trade soil carbon, incentivize farmers to build this undercover crop. Certainly there hasn't been much breeding for deep roots and microbial association in soil carbon, but there's a lot of benefits that we've taken for granted. And if there's a market for this, then there'll be a target for breeding, and we can go after it the same way we have before. Indirectly, we've actually ignored it and built smaller root systems that require more inputs. So it's opportunity. Here's one example. Actually, we started 15 years ago, and the Land Institute started maybe 30 years ago working on redomesticating a perennial wheat. This comes from the Middle East as well. It's Thinopyrum intermedium. They tried to cross it with the original annual wheat, and they could make some successful crosses, but every seed that came off was a different species, and they couldn't breed with it anymore. It was just a strange karyotype of chromosome. So they went back to Thinopyrum intermedium, got quite a diversity, and then selected again and again for plants, outliers that would have large seeds. Made some crosses between those, and that's when they reached out to us. I sent a graduate student down to Salina, Kansas, and they started to do molecular breeding. They've also sent these seeds here to Canberra, CSIRO. Phil Larkin was working on perennial wheat, and it's kind of hit a back burner, unfortunately, but maybe we can revive it now that soil carbon is so popular. This would be really perfect for soils that need improvement. So we can, there is a little trade-off. You got to invest the first year, you're investing in roots, and then the second year you can come back and large grains, and it has a lot of environmental tolerance. But breeding these perennials without genomic selection is very difficult. It's one of the reasons most of our crops are annuals, is because it's quick to make selections, and you can make phenotypic selections. But, for human disease mapping, we don't have fast generation times, and we developed this genome-wide association study framework that can be now applied to many different species with big genetic gains. So take a look at perennial wheat. You can actually drink Kernza beer. The beer market is got a high premium, and so they're using Kernza. Actually, the bread quality might be a little lower than the beer quality. So have a glass of perennial beer, and you'll go carbon into the soil. The other thing is to try to look at genetic variation underlying traits that can only be modeled. They're difficult to measure. So we might measure above ground biomass or below ground root biomass. But what is photosynthesis? And this is a topic for two large centers of excellence in Australia, translational photosynthesis and plant energy biology. For the last 7 years, they've been working at ANU to try to estimate the photosynthetic rate of leaves, the respiration rate of those leaves, autotrophic respiration. Plants need to live through the night, that's RA. And then heterotrophic respiration, RH, that's all the microorganisms that break down the carbon when the leaves fall on the ground, and the roots feeding the microbes in the soil. Those are losses of carbon. So we'd like to increase the gains in photosynthesis and reduce the losses, both in plant stress and in the microbial decomposition. So that's known as carbon use efficiency. And we need to measure the biomass, we need to measure the chlorophyll fluorescence, which tells us about photosynthesis, and then allocate carbon from leaves to shoots to roots to microbes, and partition this out. And if you look at that at a global scale, you see that photosynthesis is really massive. It's 10 times more, maybe 12 times more than human carbon emissions, which are 10 to 12 gigatons of carbon per year. This is 120 gigatons of carbon per year. But that's quickly lost. About half of it goes straight back just through the night. The plants, they make a lot of energy and they use a lot of energy. And the microbes use a lot as well. So almost 90% of the carbon that's pulled in through a leaf goes right back out. So there's a lot of opportunity upstream to work with the biological carbon cycle, especially because half of the planet is in agriculture. So we've been thinking a lot about this step, and this is an opportunity for breeding that's really largely been neglected. At the end of the day, we've got these known disturbances, land clearing, drought, fires, pests, and that does release CO2 emissions as well. Although natural systems are making up for it. Okay, so opportunities for genetic control of modeled traits. That's a real opportunity, and we'd like to add the below ground piece. There's lots of different categories of soil organic matter, carbon in the soil. Some of it is just leaf juice. It's going to be quickly broken down. There's dissolved or low molecular weight carbon is also known as dissolved organic carbon or dissolved organic matter. It's very small, low density, kind of runs down the rivers and feeds the ocean fisheries, but it's largely transient. And so much of the carbon there is lost. There's particular organic matter which is large, but it floats, and that's leaf decay. And there's not too much you can do with that. You could try to minimize it and make biochar or something. So there's opportunities. Aggregate and mineral associated carbon are different pathways. They're more stable. We'd like to increase the fraction that goes to these more stable pathways. Don't disturb the soil too much, no till. And that's another reason to go to perennials. If you get the right genotypes, you don't need to till every single year. Moving from annuals to perennials itself is a big conservation approach, and that's been limited by our ability to do molecular breeding, which has recently changed if you can do the bioinformatics. So this is what happens below ground. We'd like to take advantage of the heterogeneity rather than that being part of the noise. We know that there's hill slopes and sunny sides dry out faster, but they get more light, so your spring is going to come earlier in the north side, but you have more water on the south side. We need to account for those environmental drivers in our genetic models so that we can predict the phenotype. That's both due to the environment and plant the right seeds in the right climate. We can move away from monocultures to geographically appropriate integrated agriculture, Gaia, and fit the right, not over-fertilize, but not under-fertilize, just get the right balance so that you're working well with the microbes in the ground, that they're helping you capture low abundant nutrients and deliver those to the plants, and the plants feed the microbes in a nice balance. Of course, diseases are when that's out of balance, so we need to model and map and manage these environmental factors along with the genetics. We like to put this together into a precision regeneration station where we're capturing water, storing water, maybe recycling it up the hill during flood times, holding it for later in the season, not draining the Murrumbidgee for global plantations, but just introducing at least 10%, maybe 30% canopy tree cover on farms along the ridge lines, creek lines, contour lines, so that you can protect your crops from hot, dry winds. And also hold the soil from the floods. So, we had record droughts now replaced by record floods, and you need this ecological resilience. We can also breed trees the same way we breed crops for these dynamic environments. This is a cartoon about how we're starting to model it. Stretch this square kilometer out onto a landscape, link them together, and think about this integrated agriculture into ecosystems. Here in the middle is our favorite little third-generation sequencing machine. You guys got to use this in the genomics course. We generated a lot of data. I think you're getting a handle on how to analyze it. We convert those reads into variants, and then compare the SNP and sample matrix. Little seedlings germinating in pots under different environmental conditions, and even using drones to distribute the right seeds in the right environments. I think the last video showed you guys how to do that. Now, in this core breeding population concept, once we've identified the hybrid zones, we have identified these introgressed lines, we have identified the ancestral diversity, and we'd like to shuffle it up. We've amplified the rare things and pruned back the common things. We can put them together in a seed orchard. And this is where you grow up a gallery of different genotypes. And we've been flying drones now. We just ordered a spectral drone. It's got more than red, blue, and green. It's got a red edge in the infrared and even an 850 nanometer signal. So this is getting at the 50 shades of green, if you will, to try to look at the chlorophyll fluorescence, the physiology, these traits that can be measured non-destructively are really, or phenotypes, they're really wonderful candidate traits that we can use to see which genes control leaf color. And we know there's greens and blues, and they get stressed and turn red, or the young new growth is also red. So we can evaluate in a relatively common condition our genetic diversity sets. Now, eucalyptus is actually a big crop. There's more stems grown off of Australia than on Australia. It's a lot of diversity, of course, in Australia, but very few, probably a handful of species have been taken abroad and grown around the world. Big in South Africa and Brazil. They're actually clearing rainforests to grow eucalyptus for soft tissue paper and really nice artists. It's high quality fiber, and it grows like crazy in the rainforest. They're using tropical hybrids, temperate tropical hybrids. This is universalis, which is a whole mix of different species. India is also using it a lot, South Africa for firewood. China is growing their plantations, and the U.S. is actually interested now too. We just got a new grant to explore 700 different species with short reads. We're going to do 40 reference genomes. A lot of that's already done and in the database. Scott Ferguson's PhD, my group. And then these trials that we're putting in, we're going to sequence those as well. So population samples, they need less coverage. And we'd like to use that to establish the forests, especially this is a great green belt for Australia. China's got a great green wall proposed. India's actually doing quite a bit of agroforestry as well. And across the southern part of the Sahara Desert in Africa, there's a great green wall. So we'd like to get, 100 billion trees has been the number that I'm throwing around on an order of magnitude. These are seedlings, kind of up to 2 meters tall. And after that, they need to be thinned and shrubbed. But this is going to provide a lot of corridors for grazing and regeneration potential for Australia, and a lot of drawdown. That's a gigaton of CO2, which is more than twice as much as we burn in our country. So we have the land to reverse that. We of course need to stop burning fossil fuels, so we need to start growing. Growing is the solution, burning is the problem. So we need, in the net zero target, you need to increase the capture and decrease the losses. And land is really important for that, especially in Australia. So these ideas take time, and I'm frustrated with how slow we're moving in a lot of cases. The technology has been around for at least a decade, and even older for some of these regenerative agriculture approaches. Agroforestry is common in Africa, but not common enough. People have just optimized for single variables. So we have the computational complexity, we have the genomic technology, the phenomic technology, and there's innovators that are doing this. So we've got examples of people doing it. We need to move quickly to the early adopter stage and then scale up to agroecosystems. And I've largely thought once we get this diffusion underway, then everything will just go. You can sort of see electric cars now have kind of moved from that innovation to early adopter phase, and they're pushing hard to get to early majority. But we might have to address the laggards and the late minority early, because they'll put up roadblocks, they'll stick in the mud, and criticized from the sidelines. So maybe we need levers and rewards to bring them on board earlier, and some incentives to accelerate change. Technology alone is not enough. Sometimes it works for cell phones. Solar panels, I think we're into that early majority phase now. A lot more solar is needed, but it's really economically competitive now. So getting the barriers down is really important. And usually it happens like this. We get technology trigger, genomics, a huge wave. It can solve everything for everybody. Well, peak of inflated expectations. I have to admit that we've been overselling to a certain degree, although then the pendulum swings back and they say, well, you can't solve it. It's not perfect. It doesn't do everything for everybody instantaneously. So the naysayers get their chance to sort of shave off the over-promising. And then hopefully we're coming out of this slope of enlightenment where the truth kind of comes through. It is possible to do some things, sometime, someplace. And the economics gets behind it, the social drivers come, and technology is diffused. It becomes the new norm, the new business as usual. So genomic medicine is probably on this second slope now, and hopefully agriculture is following it. We'd like to apply it. Still early days for conservation and restoration, but it's all possible. So this is the last slide, last thought for the semester. We have a big continent and a lot of opportunities. We need to link our primary industries, which are mining and agriculture. We need to start improving soils, something for the miners to do. And this is a photograph of the Alcoa mine in Western Australia, and the Jarrah is Eucalyptus marginata old growth forests. But they clear it, they take out the aluminum, and they stack it back on contours. They put this overburden, which is the ground up forest, they put it back as topsoil and replant. And we were helping them choose the right seeds. In fact, there was plenty of diversity, so what they were doing was fine, which we didn't know ahead of time, but that's a great news story. So big equipment, big scale, big opportunity, big solutions. Thank you very much. And keep in touch. Have a great semester. See you guys.